like it's 2 o'clock, so any strong moves would be late. Hi, I'm Arielle, Dr. Arielle Johnson. I am a scientist who a lot of my grad school colleagues sort of roll their eyes and sigh at because I, instead of going on to a postdoc, went to work at a restaurant. That was Noma in Copenhagen. Now I'm also a person who's interested in stressing out plants, not so much because I'm a tomato dominatrix or anything like that, but because it gives us really cool opportunities for flavor. But so you guys all came here for chemistry, or chemistry in the title. Chemistry is what they call the central science, so it connects the world of mechanics and energy of physics with the biological world of life through the organization and interaction of matter in the form of molecules. And chemistry is a really useful tool for understanding what goes on with food, not just in the kitchen, but with almost every aspect of food. Specific to the kitchen, anything from the texture changes in meat as you cook it, or the development of crust in bread, or any of a million other things can be mostly explained by the actions and interactions of molecules. What I'm particularly interested in using chemistry for is understanding flavor, so how foods can interact with our bodies and our brains to create the perception of flavor. Foods do that with molecules, but it's probably useful to give you guys a basic definition of flavor. Flavor is what you call, or what you might call, a multisensory construct. So the 5 senses are sight, smell, taste, touch, hearing, and vision. Flavor is not one sense, but rather takes information from all the senses and combines that into a composite perception. So the 2 most important senses for flavor are what we call the chemical senses, which they studied down the street at Monell, smell and taste. So taste is responsible for sourness, sweetness, bitterness, umami, saltiness, those basic tastes. Pretty much anything else in flavor comes from smell. Smell does this in 2 ways. So when you're thinking of smell as sniffing, that's a process that we'd call orthonasal olfaction. That's where molecules enter your nose through your nostrils and then interact with a piece of tissue deep inside your nasal cavity called the olfactory epithelium. There's lots of receptors there. Those bind to molecules, and your brain creates perceptions of smells. When you're eating, the more important pathway for that is what we call retronasal olfaction. That's where volatile molecules go up the back of your throat, pushed on puffs of air when you swallow, and bind to the same receptors, but your brain integrates it into a perception of flavor. So for hundreds of years in sort of philosophy and natural sciences, smell was considered the least important sense, but it's actually the sense to which we devote the greatest evolutionary resources. About 2% of our genome is devoted to olfactory receptors. So that means that like the largest family of genes that we have is set up so that we can smell and have flavor. So that's roughly the same genetic magnitude as what separates humans from chimpanzees. So it's kind of like a big thing, and we didn't really appreciate it for a long time. Largely because the things that we smell are very, very tiny molecules in very, very tiny concentrations. So we can experience them by smelling or eating them, but to analyze them chemically requires instruments that were of a sophistication that they couldn't really be invented until the 1960s or so. So it's a sort of new and developing field, understanding the importance of this stuff. So what that boils down to, along with our genetic sensitivity and physiology, is that while mice and dogs and other mammals have a sense of smell that's quite sensitive and oriented towards survival and finding food, our retronasal olfaction part of flavor gives us a really rich world of odors that are specifically, or we're specifically designed to experience them while we're eating. And a lot of these odors come from unique biological sources and arise out of biodiversity and ecological interactions. So just like chemistry is the central science, and chemistry can be useful for understanding flavor, flavor can be interesting as a sort of thought tool as a central phenomenon between the biological and ecological world of plants, animals, microbes, and the emotional, psychological, and cultural worlds of humans and food. And so this is something I'm particularly interested in studying and working on, which brings us to this map of the Scandinavian region, because for quite a while I was doing that as the resident scientist in a place called Noma in Copenhagen. It was rated several times by Restaurant Magazine as the world's best restaurant, but what attracted me to it is that they were very interested in the research and R&D process. So the reason for that was because of a creative limitation that all of the ingredients that were used at Noma had to come from this region, which is a very cold, inhospitable region. No lemons, very short growing season, so you had to get really creative about where you're going to look for flavor. And chemistry was a very useful tool for finding that out. So when you're in the kitchen, there are, since you're applying heat to things, one of the useful ways of using chemistry is looking at flavor transformations. So a good example of this is smoking. So if you're smoldering some wood chips to smoke a piece of lamb barbacoa or some other smoky thing, you're actually altering molecules on a fundamental level and creating new ones that we experience as flavor. So this very large molecule is called lignin. That's the main structural molecule in vascular and woody plants, so trees, any other sort of land plants. And as this is heated up, the sort of big overall shape breaks down into these smaller molecules. So you can see they share some of the same structural features. Whereas lignin has no taste or smell for us. Its products are volatile, so they can bind with our olfactory epithelium. We experience them as flavor, and we get smoky, spicy, clove, and vanilla aromas from the thermal breakdown of this molecule. We also get some different molecules from cellulose and hemicellulose. So this is like the molecules that go into paper from wood. But if you break them down with heat, you get these sort of caramelized, sweet, nutty, toasty flavors. So by either thinking about the chemical composition of your wood or how you heat it up, you can alter the flavor profile that you get by knowing a little bit of chemistry. So this got me to thinking, like, there's various ways to create flavors in the kitchen, but what about starting to control and manipulate flavors before the ingredients reach the kitchen? So one really interesting tool for this is fermentation. Fermentation is when you take microorganisms, bacteria, fungi, yeasts, molds, you give them something to eat. They transform the molecules in that edible to them substance into new molecules. And this produces for us things like alcohol in beer, carbon dioxide in bread, lots of different flavors in soy sauce and miso, the sort of tangy flavors of kimchi and sauerkraut. And what you're doing sort of in concert with all of these different microorganisms, be they bacteria or fungi, is work with them in concert by creating a happy environment for them, and then on a molecular level they create new molecules and flavors for you. So these are some of the tastes that you can create using fermentation. Lactic acid from sugars, sugars from starches, acetic acid from alcohol, and umami flavors from free glutamic acid from the breakdown of proteins. And these are just a fraction of some of the aromas that you can create. So if you recall, the tastes go into flavor, the aromas create this sort of bigger and more complex picture of what's going on chemically and also what's created in your brain. So with taste, you get things like sourness and umami. the smells, you can get buttery flavors, you can get pickly flavors, you can get sweet and malty flavors, and your starting point is usually raw vegetables or grains or legumes, things that don't have these flavors to begin with. So by working with these microbes, you can create these molecules, but the more important thing for you as eaters is that you create a greater palette of flavors, which is quite useful if you are intentionally limiting the ingredients that you're working with. One of my favorite fermentations uses a mold called Aspergillus oryzae. It creates something called koji. So this is the koji room in a miso workshop in Japan. And you let the mold grow for about 48 hours, and it produces, then you mix it with soybeans and salt, and over time this creates miso. So the way that this does this is through the action of enzymes. So enzymes you could think of as the kind of molecular machinery by which life creates molecules, and in this case, the enzymes protease and amylase are produced by the Aspergillus oryzae mold. The protease will break down proteins and create free amino acids, thus the delicious flavor of umami. And amylases will act on starches to create sugars, which gives sweet tastes, but also the sort of fuel for further microbial transformations, like with lactic acid bacteria or yeasts. With something like miso, you can think of it in a sort of historical context. So traditionally in Japan, you grow mold on rice, and mix it with soybeans, and get miso in the end. But if you think of it with a little bit of chemistry, the rice is a starch source, and the soybeans are a protein source. So it's actually possible to then think about ingredients you might have at hand, like barley or yellow peas, or if we're in Zimbabwe, ground nuts and millet, and kind of swap in different ingredients that have similar chemical properties to get new flavors from the ingredients that are around you. Fermentation is one of these situations where the ultimate flavor, the ultimate success of it depends on how well you control it, and a lot of the control comes from environmental conditions like temperature, salt content, humidity, which gases are around, how much oxygen or carbon dioxide. There are a lot of very traditional technologies that have been built to work with this, whether it's wooden presses to exclude air, or temperature-controlled vats where the control comes from wrapping mats around things to keep the heat in. But you can also use more sophisticated technology to introduce these control points for the purpose of manipulating flavors. So this is a lot of what I did at Noma. You can think of the whole system as sensing and actuation. So if you want to control temperature, you can sense the temperature, send it to a small brain box, and then that will turn on a heater or a cooler, depending on how far out of whack your temperature is. So when I came to Noma, this was what our fermentation control looked like. We had an attic, and we had a heater in that attic that would turn on when the attic was too cold. We also had a broken refrigerator that we put underfloor heating mats in and used that as a control system. But when I got there, my boss, Rene Redzepi, the head chef, or the chef owner, told us, you need to build a better system for this, with no money, but just make sure it works. So we found some shipping containers. Since we could not hire someone to do this for us, we did it ourselves, cut holes for doors, and wired a kitchen and a temperature and humidity control system in. We created this sort of micro-environmental control to kind of coax microbes into doing what we wanted them to do. This is temperature control. This is, so I come from a formal chemistry training. I have a PhD in analytical chemistry. So I was familiar with many of the techniques you can use to analyze free amino acids, for example, none of which were available to me in this lab that we created. So instead, I started building analytical equipment myself. So this is like a little Arduino and LED-based spectrophotometer that we started calling the umami meter because you could then, by color, measure the amount of glutamic acid that was around. Okay, so as a segue from that, so like fermentation leads to this whole suite of flavors from relatively bland ingredients to very flavorful, often like acidic and umami ingredients. But thinking about chemistry can let you do a lot of other interesting things with ingredients. This is an example of about a dozen and a half different oils that we would make at the restaurant. So flavor molecules, as you might know, if you've left like a piece of butter in your fridge, sort of unwrapped, and then you come back to it, it tastes kind of funny. Fats are like a really, really good extraction medium for flavors. So on a molecular level, flavor molecules are more attracted to fats than they are to water. And in this lower picture, because a lot of flavor molecules are volatile, they spend a lot of time in the gas phase, it's possible to distill them out of where they were before and make a very concentrated essence. So just an example, if you're not into fermentation, of some of the ways that thinking chemically can enable you to get sort of more refined and interesting tools and ingredients in the kitchen. This is an example of one dish that doesn't use any of those tools. This is a grilled rose flatbread, and this is a dish that really got me thinking about how flavor is produced in nature. So this is a flatbread that we'd make, and then we'd gather wild roses from the beach, from the coastline. Never cultivated roses, always wild, and they'd have this like amazing, crazy, intense flavor. And so I was thinking a lot about like where these flavors came from. And sort of moving beyond the kitchen into bigger questions, what if we could somehow start controlling and creating flavor like while ingredients were still growing? Sort of the ultimate kind of, rather than farm-driven cuisine, almost cuisine-driven farming. I wondered if this was possible. So this is a slide or a picture I borrowed from Stephen Jones, because one way to do that is to look into the genetics of a plant. So either by directly modifying genetics or through traditional breeding, you can create a plant that has some more specific qualities that you're interested in, whether that be yield or shape or flavor or color. But what I've been working on a lot is looking less at genetics and more at something called phenotype. So all the DNA contained in your genes codes for proteins. So anything that your genes produce directly are proteins. Anything beyond proteins has to come from those proteins acting on something. So if you think of sugars or colors or shapes or bone or almost any other type of molecule, you're looking at how genetic expression translates through enzymes into what we call phenotypic expression. So a lot of this phenotype is affected very much by the environment. Whether that's the like local environment that a fetus or a embryo is developing in, or the sort of ongoing changing environment that an organism finds itself in. So stepping back to flavor, this means that flavor as a molecularly expressed phenotypic characteristic of genetics is deeply tied into ecology. This is actually one of the main mechanisms of how evolution works, is that some kind of molecular byproduct of a given enzyme, which the organism has really no use for in most situations, will start to confer some kind of an advantage on that organism in a specific stressful environment or in a new emergent uh, uh, climate, such as you get from climate change. The enzymes that our genes produce are able to produce a lot of different uh, types of molecules, and these are created sort of on the fly in response to environment. And this is especially the case for plants. The rest of my talk is mostly going to be about plants, because plants have, I think, the most diverse array of flavor molecules to use in the kitchen. So plants are what we call sessile organisms. They're not able to move or walk away or fight back if they're being attacked. Whereas organisms that can move have developed different strategies like that, plants interact with their environment mostly through chemistry. So for things like dehydration, ultraviolet exposure, heat, insect predation, predation by pathogenic fungi, those first 5 are stresses. All of those situations are mediated by plants using chemistry. In the case of dehydration and UV exposure, when the first land plants evolved from algae, the molecules that they used for those two things were the first adaptations that they were able to evolve to come onto land. Because underwater, you don't have UV exposure, you don't have to worry about drying out because you're surrounded by water. Then once like insects and fungi came around, you started seeing plants developing different defensive compounds to deal with these things. There's also the realm of chemical situations where you want positive interactions between plants and their environment. So whether that's attracting pollinators or getting herbivores to eat the fruit that they produce so that their seed can be spread. That's a whole other suite of chemicals that are produced by the plant and that we can taste and smell and use to our advantage if we know a little bit about chemistry. So all in all, this means that plants smell like green spirit. This is actually, I wrote an article for Lucky Peach about this, but the really interesting thing about a lot of these molecules is when you hear like defensive compounds, you think, oh, that must be like bad tasting. And in a lot of cases, that's true. So alkaloids like caffeine or capsaicin, which is spicy, or some of the sulfur compounds in garlic and horseradish are directly intended to like deter humans or animals from eating plants. Obviously, it doesn't work because we grow these plants specifically for those compounds because they're delicious. But there's also a lot of volatile components, so flavor molecules, that in the plant were selected for by evolution to deter an insect, or maybe if they're being eaten by an insect, attract the predator of that insect. So things that have nothing to do with humans that we find delicious, which is quite exciting. So yeah, here are some examples of those compounds. Caffeine is a pesticide. Allyl isothiocyanate is a lachrymatory compound that is a delicious component of wasabi. Camphor, which gives a kind of piney quality both to pine trees and to other herbal species, is intended by the plant to protect from drought. And there's various others. Citral. The red color that we value in red wine comes from molecules like peonidin, which is intended as a kind of sunblock by the plant. Coming from this place where we're thinking about, or I was thinking about how to control a microbial ecosystem by looking at what they responded to and using those as control points, these evolved responses become new control loops. So if you want to create a plant with a lot, or if you want to use a plant that has a lot of this pine flavor, you can intentionally dehydrate the plant to the point where it has to protect itself. Similarly with UV exposure, applying heat, or applying some version of predator insects or pathogenic fungi. So that has brought me to my current job, which is at MIT. That sort of ominous pink glow is our lab. It's an indoor farming lab. So this is the Open Agriculture Initiative at MIT. The current research of the whole group is sort of taking a more engineering approach to what I just described, because there are a bunch of engineers, is looking at biological systems as a system of inputs and outputs. But, so, like I said, I have to do things myself to get things done, so started building experimental environments to do some of these intentional flavor controls on plants.
So you need a little more money this time.
A little bit, yeah. Yeah, so we're able to get a new shipping container instead of a used shipping container. And it's inside rather than on the parking lot. So yeah, so like a lot of what I'm looking at now is stressing out basil, which is a very tasty plant and a good model organism because it produces a really wide range of flavor compounds. And then applying some of these situations that it's evolved defenses against to cause it to ramp up its defenses and create something that's tastier to us. So these are some of the main defensive molecules in basil. Estragols, kind of tarragon-y, camphors, piney, eugenol tastes very strongly of cloves. There's also some woody and herbal and sort of floral compounds going on there. So we grow the basil. We are currently looking at things like supplementary UV light. UV is like creates a lot of chemical stress on all kinds of living organisms, so it's a pretty easy way to stimulate a lot of these chemical defenses in plants. I tag them and harvest them, and then analyze them using something called gas chromatography, mass spectrometry, which is basically the tool we invented in the 60s to start looking at volatile molecules, which were invisible before. So yeah, by applying different lights and different sort of photo periods of ultraviolet, we get these really interesting interactive responses that we're still sort of teasing apart, where like different elements of like the visible light spectrum will create sort of interactive effects with like a 17-hour UV exposure versus a 4-hour one. But basically, we're able to like really amp up the flavor production in basil. And kind of the whole point of this, beyond making cool pink sort of robot things, is to collect this data and share it openly with other people that are interested in either experimenting with plants or growing them themselves. So a lot of our focus now is on basil and the flavor compounds of basil, but we're very interested in looking at the kind of diversity of chemistry that plants produce and creating situations where we can either manipulate that to our advantage or study it further. So, if you have a contained indoor environment, you can simulate what a climate under climate change might be like in 15 years, and then try to figure out different conditions you can change in that, such as like water or supplementary things, or test out new breeds of plants to try to figure out like what is going to grow in an environment that doesn't exist yet but will exist in the future. We're developing sort of an open database of this data. And what's really exciting for me is that like that doesn't necessarily have to be confined to plant data. Really, what I see in the future is some of this collaboration between restaurants and institutions like MIT leading to databases where cooks can share all kinds of information and knowledge with each other, sort of parallel to this more closed, paywalled academic publishing system. So yeah, I was intending to leave some time for questions, and I think I have. So if any of you have any questions or anything, please. Hi, yes.
Yes, I mean, you talk a lot about like volatile compounds that we can't actually see. So I mean, like when you interact with a plant like basil and you smell it, it's actually like giving off molecules, or it's
Yeah, so in a plant like basil, most of the basil smelling molecules are stored on like hair-like structures on the underside of the leaf. And when any of those are like disturbed, even by like ruffling wind, molecules are released and they make their way like into your nose, and you actually smell a physical part of that
yeah,
plant.
but it's something completely invisible, it's just floating in the air.
Yeah, I mean, like the general concentration ranges would be in the like parts per million or parts per billion. So if you have like pure eugenol, that's one of the molecules in basil. I mean, you can get like quantitative amounts of it. Like if you get a gram of it, you can see that, but it's also like extremely unpleasant because it's like way more than you'd ever smell
Yeah,
in nature.
overload.
Yeah, but I mean that's why we have the sensory system of smell, is so that we can have detection of compounds that like way below what vision can see.
Yeah.
I guess.
Hi, two questions.
Mhm.
One, I'd like to know your favorite dish that you've eaten at Noma. And two, the creative process for a restaurant like that that's
Mhm.
employing chemists and chefs
Mhm.
and farmers.
Yeah.
The creative process for the restaurant industry is usually some guy that doesn't know anything about anything and comes up with something that tastes really good.
Mhm.
What is the general process for menu development at Noma with all of those different ideologies?
Let's see. First question. I don't know if I could pick a favorite dish. I mean, the rose flatbread that I showed you was like really amazing. One of my other favorites was made by a chef who's no longer there, Rosita Sanchez. So she was the pastry chef and now owns a taqueria in Copenhagen. But it was a potato and plum dessert. So like sweetened like white potato, and like plum kernel cream, and then like an aromatic spray made out of fermented plums. So yeah, it's a place very dear to my heart. But so back to your question about the creative process. Were you at the sessions yesterday? Okay, so for those of you that were not at the earlier talks yesterday, Pilar Opazzo was talking about the creative process of LBE. And LBE was a restaurant that closed in 2011, but basically founded modern experimental cuisine. So was really sort of the forerunner of places like Noma. And her sociological study of LBE detailed the sort of obsessive amount of documentation that goes on in LBE's creative process. I highlight that because Noma could not be more different from that. It's one of the most sort of chaotic creative environments I've ever been in. But the sort of design there was to bring together people like me who can do experiments but also communicate to a chef, and chefs that can also think very experimentally, chefs that are like extremely creative and can't really maybe not run a kitchen but are perfect in a test kitchen, and to kind of throw us together and have us sort of talk to each other ad hoc. I mean, I think probably what a restaurant that's trying to develop its creative process might take away from that is something in the middle of the two. We were very ad hoc, but we developed eventually some systems for sharing information. And I think even things as simple as like Dropbox recipes or taking photos on Evernote, and everyone was subscribed to the same Evernote notebook and could see like at least some of what was going on in other spaces. I think what you're seeing now that's really interesting and exciting is that like restaurants aren't just confining their creative process to the one restaurant itself. So a lot of chefs are communicating with each other about things like fermentation or weird varieties of vegetables or flowers or fruits or something like that. Instagram has been a really powerful tool for like enabling that type of communication, for better and for worse in some cases. But like my hope is that as we see like more people working on the sort of technical aspects of food that care about restaurants and farms and things like that, that that dialogue develops in the same way, kind of extracurricularly to any one given restaurant. I mean, because it's super expensive to have in-house. It's crazy expensive. Like, no one can afford to do it, but there's enough people around that are doing interesting things and documenting it and are willing to share, which was not really the case even 15 years ago. That I think this new ecosystem of ideas is kind of becoming like an outside the restaurant sort of cyber test kitchen type of thing. Sorry, I talked for a while. Do we? Hi.
Hi. Are you doing any of your ag testing actually out in the fields, or is it all inside?
Currently, we are inside. My project over the next probably year is figuring out how to deploy a lot of these things in the field. I mean, outside, it's not really feasible to like have really tight control of like soil chemistry and things like that. But even just collecting data sets about like what happens environmentally in a field and then like relating that to the plants that come up. I mean, other people do that. I like didn't invent doing that. But for the most part, the data is very closed. So not a lot of farmers can benefit from it. So the hope is to like leverage this sort of open sharing of data so that more knowledge can be spread from like indoor research farms to farms outside. Oh, in the back?
So, like, what you're doing has been kind of like done in a way with like grapes, like winemaking, right?
Yeah.
Like stressing up vines,
Totally.
and trying to bring out different flavor profiles. Do you think are you like with basil in particular?
Mm-hmm.
Like you say, it has like tarragon flavors or clove flavors. Is your goal to be like, I want to make this taste more like tarragon or more like clove, or I just want it to taste more like the basil that it already is? Like, do you feel like you'll be able to manipulate like individual flavor profiles within plants based on like more UV or different types of bees or whatever you come up with?
Yeah, I mean, more as like an academic question. I'm interested in that, and I'm working on it. I mean, I'm not sure if it'll be feasible large scale, but like, especially since like the tarragon and clove flavors come from a different biosynthetic pathway than the like more herbal, piney, floral and woody flavors.
Right.
I mean, one is just interesting on like a biochemical level to see how different stresses activate those pathways. But
Right.
like, yeah, it's also would be really cool, and we're looking into how to tailor that.
Because it seems like the wine industry would want to reach out to you
Yeah.
to say like, we want a Cabernet grape that has more of these qualities.
Right.
Or,
I mean, and if you are into something like that, I mean, you can think of this whole ecosystem as like sort of fictional terroir. So, I mean, it's the same mechanisms as produced terroir. You have a certain set of genetics, and wine is an excellent example since, for example, Pinot Noir and other varietals are, for the most part, all clones of each other. So you can produce the same clone of a given grape, so the exact same genetics in two totally different places, like the Russian River or France. The phenotype that you get is completely, the differences are completely dependent on environment. That is what you see in the flavor. But yeah, there's all kinds of sort of parallel and fictional terroirs you could create by doing the same things. I don't know anyone who's doing that, but I'm sure they'll find me. I actually did my my graduate research at UC Davis in the wine department, so yeah, wine science is pretty close to my heart. I think.
So opening that up on a larger
Yeah.
scale, do you see this being more in the role of a cartographer or more of a conquistador? Are you looking at this as a mapping project, or are you actually looking to try to change things?
I mean, I would say like a mixture of both. I suppose it might be more like curation than cartography or conquering. I mean, the thing about flavor molecules is that like humans, except for like sweetness, can't sense nutritional compounds directly. Um, so flavor is what we use as a proxy for like nutritionally dense uh, plants and other food stuffs. So a lot of the sort of specialized metabolism that creates aroma compounds gets turned on at the same time to create like antioxidants and other phytochemicals. So, you could view it, or I often do view it as a project to try to make as plants that are focused as much towards their specialized metabolism as possible. And, they are more delicious and as a side effect also more healthy for us. So I think like that kind of looking at flavor that way is kind of like behavioral economics is what in some ways makes the most like salient argument for like ecological protections and organic farming because it's actually more stressful on the plant and biodiversity. So it's not, I don't really view it one way or the other. Yes.
You talked a little bit too about evolution, and I wonder, like, when you're doing some of these projects on basil and you're giving it these stressors, is it potential then that these basil plants will produce new strains of basil and that it'll evolve and grow depending on?
We haven't actually let it go to seed yet, but
Okay.
So applying these stresses to reproducing plants is kind of exactly how you do seed breeding. So you put the seeds in the environment that you know that they want to grow in, and then of the different sort of phenotypes that result, because you're going to always have like random genetic variation, you artificially select the ones that have the properties that you want. So it would definitely be theoretically possible by applying this sort of special like UV light, or we're also working with a compound called chitosan that's developed from shrimp shells that confuses the plant into thinking it's being attacked by either insects or fungi, so like ups its defenses. You could create an environment where, like, in successive generations, they get better and better at like defending against these things, and then also make more flavors. Yeah, evolution is exciting. So, yes.
Is there such a thing as a periodic table of flavor?
No. I mean, there's some like proprietary databases of like all volatile molecules, or many volatile molecules, and like what their flavors are. But there is not, to my knowledge.
They're proprietary too.
Oh, I mean, if you wanna pay like $6,000 a year to access it, you can. One of my hopes from this is to open that information up. There's actually a lot of those relationships like we know already, or like science knows already, but it's not in something like a periodic table. It's in chemical data that only exists as a table in a PDF that's behind a paywall, which to me is very sad and bad. And I'm hoping that the progress of like this whole field goes more towards opening up and indexing and cross-referencing and making it more accessible to people that care about flavor molecules. Oh, hi.
Hi. So I guess I'm curious if the compounds that you're studying that you're trying to increase plant defense compounds are obviously some of the most bioactive compounds these things produce. They're meant to interact with other species and have an effect. To what extent do we know? What percentage of these is the actual physiological effect well known and understood? I know that it's caffeine, vanilla, and all these things are known, and then you have a lot of the stuff you're working with, which do we know what it does? I'm not.
I think a lot. You're right. A lot of it is unknown, and in terms of biological activities, like that's actually a big project of like drug companies is to do screening assays on all kinds of wild plants to try to find like new molecules with new activities. So it's like an ongoing study of like metabolomics and sort of structure-function relationships, but
And that's mostly also proprietary, I'm assuming.
a lot of it, yeah, unfortunately. Yeah, I mean the science around food has really like developed alongside the sort of industry of food, so the process is designed generally more to benefit that than to benefit consumers, unfortunately. I mean, I think it's slowly changing, but yeah, I mean, open data would be a big. Hi.
So part of the hospitality industry and cooking is having diversity, right? So
Mhm.
people go to Noma
Mhm.
for the diversity of the, for lack of a better thing, like it's basil versus the basil we have here in Philly.
Mhm.
The example with wine, I know the wine industry went through a problem where all the grapes kind of started tasting the same.
Yeah. Yeah.
With our industry being very cyclical, how does your work differ from, back in the late 70s and 80s where we started mass producing the best tasting pork, for example, or best tasting chicken?
Sorry, I'm gonna stop you because I don't think what we were doing then was creating the best tasting pork or chicken. I think what the pork and chicken and wheat and corn and other commodity organisms that were produced then were the like highest yielding versions of that. So yeah, in fact, like most products for breeding or most programs for breeding organisms do not look at flavor and have not looked at flavor. Stephen Jones being a notable example or counterexample. Yeah, so I think the world would be a lot more interesting if people are trying to breed for maximum flavor, actually. But sorry, not to cut you off.
No, that makes perfect sense. I was just starting thinking full picture.
Yeah.
And how your work, I don't know.
I mean,
there was chemists back then that thought the same thing and kind of evolved to what it is.
I mean, I think like a lot of chemists that were employed in this process would like look at kind of basic things and not really flavor. I think flavor is one of the strongest arguments that we have for biodiversity. Because we've evolved this system for millions and millions of years, and the conserving anything through evolution takes a lot of like energy and effort. So if we are able to smell millions of interesting molecules, we should probably do that.
I agree. Thank you.
Yeah. I think they probably want the room for the next talk. So if we have like any other questions, I can talk to you. But thanks so much. Hi. Oh, okay. Oh, great. Cool. Yes, I mean, yeah, oh, okay. Yeah, there's FlavorNet and also Ferrobase and Cookery are like pretty okay for that kind of data. I actually like use it a lot for like basic stuff. Yeah, no, they're great. It's like a really good.
I love the science.
Oh, you do? Oh, not at MIT, but yeah, I did.